So here at Loughborough University, we're creating mini tumours inside a bubble. And we're doing this because we want to improve the way that we test anti-cancer drugs before they get to human trials, so that they can be more effective and we can reduce the number of animals that are used in research. I've started the process of making the bubbles now. We would have the cells inside the jelly material here. It's being pumped into the bottom of this system, and then it's being pushed up through this membrane, which has lots of little holes in it, which basically makes those droplets. And you can see here, it's very cloudy. That means we're making the bubbles. Once they're made, the cells will already be inside. We'll take it over to the microscope and we can have a look. So my technique is using something that can be very easily scaled up. So we can go from a system that I'm using in the lab to something that can produce thousands and thousands of these little models in a few minutes. Most of them, 97% of cancer drugs, actually fail. They don't work when we test them in human trials. And that's because the way in which we're testing them before we get to humans, so in the lab or in animal tests, they're actually not very good, and they're not very representative of what actually a human tumour is and what it contains and what's around it. So my tumour in a bubble tries to kind of recreate that very, very complex tumour environment in a lab setting, so that we can use this to test whether potential drugs are going to be effective at killing the cancer cells. And hopefully this would be a better kind of system of testing those drugs than the animal models.